I'll kick off the Q&A session um, with a couple of general questions. So we had a question for the entire panel. So whoever wants to go ahead and answer these questions, or if more than one person has an answer that they want to provide. The background in cowpea, we're talking about concurrent protection. So in cowpea, it has been shown that infection with a less severe strain can provide protection against a more severe virus. Have any of you in your work with cucurbit viruses or are any of you aware of research looking at mixed infections where the presence of one virus or strain appear to suppress or limit infection by another virus? And if so, do you see any potential relevance of that phenomenon for breeding or disease management?
I can take that one if you want me to, even though I
Go
wasn't
for it.
speaking to the. So we've done quite a bit of work looking at competitiveness of different viruses and mixed infections. This is largely because we have so many cucurbit viruses that are occurring together in various parts of the world. There are a number of ways that can happen. There are some viruses like those that many of those that were described today that are moving systemically throughout the plant. And I'm not speaking for cucurbits, but in many of those studies that have been done, people have looked at where those viruses are located. In some cases, they can co-infect a cell. In other cases, you'll find that one virus may infect a particular cell of a plant. And because of that, you'll see that another virus may be in nearby cells, but not in that cell. So there is that type of an interaction that can occur with certain types of viruses. Some of the other viruses we have are more phloem limited, and you can see many viruses occurring together in those types of situations. That being said, even when you see those types of co-infections, you'll often find that one virus may do better, say if it gets a head start, gets in earlier than another, that may dominate the infection. You may find that viruses that come in later may not compete as well. This is particularly true with viruses that are related to one another. Other dynamics, and others might want to chime in on this, you'll see synergistic interactions where one virus may make the symptoms on a plant more severe, and sometimes that can also lead to an increased virus titer for one or the other virus. So the dynamics are really interesting. Sometimes even unrelated viruses or very distantly related viruses, you will see interactions, likely because of the way the plant reacts to the infection itself. I don't know if anyone else wants to chime in on that.
All right. We'll move on to our next question. So, two questions talking about all of the viruses that were discussed today. One of them is, or maybe not even just today, but are any of these viruses or those transmitted that affect cucurbit crops, are any of them transmitted by pollinators? Any knowledge on that? And I guess I should introduce Dr. William Wintermantle. His bio is on there. He is serving as co-host today and is co-chair of the EVCWG. Any known pollinator transmission? Sudeep, I see you took your
So
volume.
it it may not be a pollinator, but I would go more towards pollen transmitted viruses. So it will be the pollen that will be transmitting viruses through pollinator. So I'm not very much aware, recalling anything that is specifically pollinator transmitted viruses, or bee if I say in this case. But if any pollen transmitted viruses are there, pollinator can be a vehicle for that.
All right. And we have, thank you. The next question that we have for any of the viruses discussed today, are you seeing selection towards virus isolates that cause less severe symptoms, similar to what is being seen with potato virus Y in potatoes? Any thoughts or observations on that?
I can take a stab at that one. I think for a lot of these viruses that we discussed today, they have especially like CMV, there's a really wide host range, right? And so like for the more generalist the virus is, I think the more difficult it would be to detect or even try to measure any selection or evolution towards a certain trajectory in the field because it's moving through lots of different hosts, right? So I'm not aware of any studies that have documented that for the viruses that we discussed today. But it would be interesting to do that. One that we're kind of looking at from that perspective is the Cucurbit aphid-borne yellows virus, because that one is more specific to cucurbits and has a narrower vector range. And so we're actually studying that process to see if there are changes in the proteins that help it infect the plant over time. But no data yet from that to share.
Great, thank you. Okay, we had another question. Of all the viruses talked about today, and I guess this would just be a summary, which ones could be seed-borne or not? And so I know we had that specifically. Dr. Vlad talked about that with squash mosaic virus, and Dr. Bagg mentioned that with tobacco streak virus. Has tobacco streak virus been proven or demonstrated seed transmission in cucurbits, or was that in other crops?
That was in other crops. So we detected TSV only once, and after that, since last 6 years, we have not able to detect it again. So it need further studies in case of cucurbits.
So when you detected it, what kind of impact like how widespread was it through the field? What kind of impact did that have in your
It was
observation?
one county and maybe a couple of fields.
Okay.
In the case of CMB, it depends on the crop. Yeah, there is some sort of seed transmission in solanaceous crops and in some veggies but in the case of cucumbers or cucurbits, I think it's less common. I've seen some studies where they found it in the seed coat, but it's not been, I guess, sufficient to transmit the virus. And because normally you will have the virus in the embryo to be able to have a complete plant infected, but yeah, it's more rarely in the case of cucurbits.
And
I did, go ahead.
Oh, sorry. For ZYMV, there is some seed transmission, but in cucurbits, but the resulting infections are often cryptic, which means they're not showing symptoms. So it wouldn't necessarily be something that you can detect in your field with symptom expression, which is interesting, but it's not a very high efficiency of transmission.
And I did share the link to our cheat sheet of cucurbit viruses in the US that does list seed transmission on there as well. And we did get a message in the chat. There is a paper reporting seed transmission of CMV and pepper that was by Ali and Kobayashi. So that was in pepper. I just wanted to share that. All right, I think those are some of the general questions. So we can move to the specific questions. So Dr. Mock, you are up. Referring to CABYV, cucurbit aphid-borne yellows virus, do we know if it infects peppers?
So as far as I can tell, no. From reports, published reports. However, with our work on looking at infections outside of crops, We have found it in related plants that are in the same family as pepper. And so I cannot rule it out as a possibility based on that. And it also is transmitted by Mysis persicae, which is a major aphid pest in pepper. So this would definitely be something interesting to follow up on. If it is present, I suspect that it's not economically important. Otherwise, usually the ones that are the main issues, we would have seen them recurring to such an extent that we would take the initiative to get funding and identify what this virus is, and so on and so forth, right? So like we see in cucurbits, where it's not typically a major issue, at least in the US, it may be present, but not causing enough disease to be a major problem.
All right. Another one for you. How many hours are required by the aphid to acquire and infect plants with CABYV?
Great question. Typically for viruses in the genus that CABYV is in, they need at least an hour if everything is perfect and they get right into the phloem to acquire some virus. The longer they feed, the more virus they will acquire, right? So if you let them, if they're on there for a whole day, 24 hours, they're probably more likely to be able to acquire enough virus than if they were just on there for an hour. But they can acquire enough in as little as about 60 minutes to 2 hours to be able to transmit. And likewise with inoculation, it takes about an hour for them to get into the phloem. And so that's actually a much more rapid process, right? That transmission can occur even faster than enough acquisition to make that vector able to transmit.
All right. You are out of the hot seat for now.
Great.
We are moving on to Dr. Escalante. All right. Regarding cucumber mosaic virus, could other viruses mask CMV infections? This was pointed out because it is less commonly detected in Puerto Rican cucurbit fields compared with papaya ring spot virus and zucchini yellow's mosaic virus.
It could be. There is a term we use in virology. It's called superinfection exclusion, and there are many viruses that will cause more severe symptoms or will mask the symptoms of other viruses, and that makes it very challenging. So that's a possibility. I don't know of any specific studies that have been done studying exactly that effect, but that's very common with mixed infections. I don't know if anyone can chime in on that. I don't know, but that's based on the information I have. That's a very high possibility. That's very common with many plant viruses.
All right.
That makes it very tricky with diagnostics sometimes also. That's why we have to have a wide array of tests to be able to see what we have in there.
Yes, it does. All right. Again, regarding CMV, is bitterness related to plant genotype? We're wondering if there might be plant varieties that have acceptable quality, potentially under still with it being infected.
That's an interesting question. So bitterness is basically a inherent natural effect of many cucurbit crops, and that's if you go to the wild ancestors or wild types of cucurbits, you will see that the fruits are actually bitter, just like and that's a natural mechanisms that plants have, especially to protect against insect pests. There is a gene that will trigger high amounts of cucurbitacins, which is a compound that is bitter, and that's usually will repel insects. So I will say it is genotype dependent. There might be some genotypes or cultivars that might have, probably because of selection, some sort of a higher ability to express these genes that are producing cucurbitacins, and so some cultivars might produce more bitterness than others when it comes to infestations of insects, and so that might vary. So, but like I say, this is a natural effect, and it's so breeders actually have been selecting for elite lines that are well, that happened a long time ago during domestication. That have mutants for these genes that produce cucurbitacin that will have less bitterness in the fruits. So that's definitely genotype dependent, and some varieties might be more prone to produce that after infection of the virus. That might not be necessarily related to the virus, but actually to the infestation of the insect.
All
I
right,
don't know
you
if that
mentioned,
answers the question.
I think so. So you mentioned breeding. Can CMV resistance levels vary depending on geographic location or the mixed infections?
I think it definitely can vary. Just like CMV has a broad range of host. It's very broad, but it's an RNA virus, meaning that it's more prone to mutations. So, and it is widespread throughout the world. So there is definitely a lot of different strains. Some of them being probably more severe than others. So that itself is gonna make it vary a lot. So in terms of geographic location. And in terms of mixed infections, it is the same. You know, there are many varieties that of course have been bred for resistance to these type of viruses, but might not be bred for resistance to other viruses. So that's where we have some of these gatekeeper genes, CMB, for example, that basically I believe CMB1 and other genes. I don't remember. I think CMBPR41, something like that. Those are very common genes for resistance to CMB that inhibits the entrance of the virus through the phloem systems. So the virus will stay just local at the bundle shield cells before entering the phloem, and that is basically the natural effect that these genes have. But zucchini yellow mosaic virus, for example, might not be being they might not be kept in that stage, so they will open these natural pathways that the viruses have, and then CMV might just hitch a ride and then go. And so it's very complex, and it depends of what viruses are in the mixed infection, and so it definitely depends. I guess the short answer is depends on geographic location and also what type of virus are in the mixed infection, and they definitely play a role in terms of resistance.
All right. Last question for you. Speaking of mechanical transmission of CMV by tools and farmers' hands, how efficient is mechanical transmission in the field? And does the virus need to get inside the plant sap to infect, or does it just need to land on the outside of the outside surface of the plant?
So the virus needs a natural opening, or I guess an opening to be able to enter, especially when we are talking about CMV. It needs to get to the phloem, especially to be able to be transmitted systemically. So I will say it depends on what kind of mechanical transmission we are talking. Like I mentioned in the presentation, when it's harvesting, where you basically cut a certain tissue of the plant and you have this tool and you move it to the next plant, that's you create an injury there. So there is definitely be a direct exposure of that sap that was infected with the virus, and that can be transmitted. And so but you can have sap, just put it there, and if any natural injuries, like because of wind or any other type of natural entry that might be created, that's a possibility. But I think I don't know if I don't know, maybe Carrie might expand on that, or
Yeah. Yeah, the key is that the cells in the receiver plant have to be alive and not damaged so much that the
Mhm.
virus can't establish in the cell and start reproducing and replicating itself. Right. So if there's something that's really damaging to the plant where that cut takes place, and it's like a super violent rupturing of cells, it's actually less likely that the virus would be spread that way. But if it's just sort of like you have sap on your hands and then you rub it onto a leaf gently so that the cells are maybe like punctured a little bit, but they don't die, then the virus can get in and infect. And that's how we do mechanical inoculations in the laboratory, right? I've often have to, sit there and train students on how to do it right so that they don't do too much damage to the plants when they're transmitting, because if they do too much damage, the cells get damaged, then the virus can't establish. So it's kind of a middle ground there for that mechanical route.
Yeah. But from the practical standpoint, I will say, there is a high possibility of transmission during especially cultural practices in many cucurbits where you have to handle the plants. And so just be careful on that part.
All right. Moving on to squash mosaic virus. Do we know about the viral particle distribution of squash mosaic virus in plant tissues, including is there any variation that we are aware of that occurs by the host?
So most of the work I came across was actually looking at the distribution and developing seedlings into plants, and in those cases they found that it was systemic throughout the plant and pretty much in all plant tissues except for pollen, and that the highest titer or the lowest titers are actually in the roots and in the hypocotyls, but the highest levels were in cotyledons, and that typically led to the highest levels in the developing plant. Variation. I know there are some local lesion varieties of plants out there that are local lesion for the virus itself. So.
All right, and another one on squash mosaic virus. Do we have a hypothesis on why the melon strain versus the squash strain of SQMB may have a different impact on seed transmissibility?
I might punt that one to a virologist. I did not come across anything. I would assume it has something to do with the movement proteins in these different viruses and these different strains and how they interact with the plant, the different hosts themselves. I know most of the work was focused on really trying to find improved diagnostic assays that would basically detect all of the strains at once for seed testing purposes. So. Carrie or Bill or Sadeep, you have any? Not something I came across in the literature.
Yeah, I'm not familiar for that virus, what the mechanism would be for more or less seed transmission.
And perhaps that's something maybe we can try and address ask during the next webinar when we have some seed transmission virus speakers. All right. If anyone has any remaining questions, I will say go ahead and put those in the chat. We did have a question related to, sorry, not in the chat, in the Q&A box. We did have a question or two related to melon severe mosaic virus. Have there been any reports of resistance screening on cucurbits? None that I am aware of. And the other question was, I mentioned that there was variation in melon severe mosaic virus symptoms, and we said that may depend on the host. The question is, does this suggest the possible involvement of host resistance factors? I would say that is a potential, but there's not enough information known about it yet. This is just some quick anecdotal observations in the field. I hope to look more into that with different varieties just to see if we can get a better catalog of those symptoms, but I just think there's not enough information right now to look into that. And let's see, looks like we had a question. Is there any information about watermelon vine decline transmission by whiteflies? Is that a main problem currently in Florida or other southern states? And unfortunately, since we are at the end of our time, and that was not a focus of this week's webinar, I will say that we addressed squash vein yellowing virus with watermelon vine decline in the last webinar. And we will have, hopefully in the next couple of months, a fact sheet on squash vein yellowing virus that both of those should address those questions and will be available online in the next few months. One other quick question. How do the findings affect quarantine regulations for import of cucurbit seeds into the U.S.? Are there any regulations already put out there? Does anyone want to try and tackle that? Or let's see. That may be something again to consider next time when we focus on the seed transmission aspect of it.
I would
So-
recommend we punt that till we have a panel that's focused on seed transmission.
Rebecca did unmute me if you want me to chime
I
in.
did unmute Samantha. So Samantha Thomas is another one of our EVCWG members, and I know has information with that. So I unmuted her to allow her to speak if she wanted to address that.
So the question is on, are there any import requirements on cucurbit seed? Is that the generality?
How do findings affect quarantine regulations for import of cucurbit seeds into the U.S., and are there any regulations currently in existence?
Okay. So today, for cucurbit seed, all that would have to come in with the phytosanitary certificate. The declaration of the species would have to match what's in the box. But are there any specific additional declarations for pest required? There is not today. While CGMMV is considered, I think it's absent and under official control, there is not an import requirement associated with that. As Gary shared, SQMV is known to be in the U.S. A lot of its early studies were actually from U.S., so there's no reason to regulate that. And then next week, or in a few weeks, Olivia will talk about MNSV, but that also is present in the U.S. So no, there's not any requirements today around specific pest. Should there be an increase, or should there be a new seed-borne virus that occurs, that is something that the USDA could look at. They could do a pest risk analysis and then decide, the 6 seeds are risk threshold. We believe seed should be mitigated as a pathway, and they could add that declaration in the future.
Thank you for joining. And with that, I will say thanks to all of you. Again, thank you to our speakers, and thanks to all of you for joining us.